name is Haonan. I'm a PhD student at Helmholtz Center for Infection Research, Germany. Today, I'm going to share my poster with you, which is on potent small molecule inhibitors as pathoblockers of alpha hemolysin of clinically relevant Staphylococcus aureus. As aureus is a constant threat for public health, mostly because of its multi-drug resistance. The strategy we want to use here to fight this microorganism is to find so-called pathoblockers, which aim only to disarm but not to kill this pathogen in order to prevent development of new antibiotic resistances. As aureus produces various toxins, but we are only focusing on alpha hemolysin because the disease we mainly want to tackle is the S. aureus-induced pneumonia. And alpha hemolysin is believed to be the major cause of this potentially life-threatening infection. Talking about alpha hemolysin, also called HLA, it is a pore-forming toxin, and during infection, it will undergo a heptamerization process and some conformational changes on the side of the host cell membrane and eventually build pores in the cell membrane. And our goal is to find compounds which can block the activity of HLA at the early stage of the potential S. aureus infection and therefore prevent S. aureus-induced pneumonia from happening. As a part of our previous research, my colleagues discovered and optimized first-in-class small molecule inhibitors of HLA by high-throughput screening in collaboration with Lead Discovery Center in Dortmund using recombinant HLA and cell culture-based calcium influx assay. And eventually, they found quite a few potent inhibitor series, but we only selected 2 frontrunner compounds from our QDS inhibitor series because these 2 selected inhibitors are the most promising inhibitors in the screening assay, as shown here. As you can see, their EC50 values were proven to be in low nanomolar range. And going on with the current part of the project, we really want to validate these 2 selected QDS inhibitors in an in vitro infection model in order to mimic real infection scenarios and also in more clinically relevant settings. So my story really started with 116 clinical isolates. And first, we looked at the diversity of these isolates. Based on whole genome sequencing data, we could really identify a high diversity of our 116 clinical isolates in terms of their genome types and also the HLA types they carry in their genome. Also, using immunoblotting, we were able to assess different HLA secretion levels among 116 clinical isolates in our infection models. Finally, what we ended up doing was to use LDH release of A549 lung epithelial cells as an indicator of HLA-induced cytotoxicity, and also completed a dose-response study of these 2 selected QDS inhibitors with clinical isolates, which secreted a detectable amount of HLA in our infection models. In the end, we could successfully prove that our 2 selected QDS inhibitors, H052 and H831, are broadly active among diverse clinically relevant Staphylococcus aureus in nanomolar range and also independent of HLA types they carry. These are already very promising results, which strongly indicate that our frontrunner compounds H052 and H831 have a great chance to be used as prophylactic agents for the risk patients to fight S. aureus pneumonia. However, some highly cytotoxic strains did not secrete HLA in the infection model and were therefore not inhibited by our compounds. And thus, we are not completely satisfied. And now we are establishing a proteomics approach in order to identify different S. aureus toxins in our infection model. And we really want to understand what could be the reason for that, and also which other toxins could play a role in inducing high cytotoxicity in our infection models. And from the preliminary data, despite a huge number of human proteins, we could successfully identify Staphylococcus aureus proteins in our infection samples. So now we can't wait to see what it could lead us to find. Thanks.